So you always start with trying to find a topic. So these are often used, or the resources that are often used are NCBI, repositories like PubMed and bioprojects and gene expression omnibus. These are all the and SRA. These are all the repositories where you will find publicly available data. So you can start with PubMed and then look for or search for some of the topics that excites you. And for example, let's say that if I want to work on SARS, so obviously SARS and coronavirus, these topics are famous for the last 2 or 3 years in the pandemic era. So once you understand different questions and different reviews that are done on SARS, maybe you will find a very interesting topic. So let's say this topic of understanding the imbalance host response to coronavirus infection, so is a one that excites you. So what do you do? So you start to find data sets that are either associated with this publication or a data set that are associated with host response to SARS infection. So if you find data sets that are associated with this publication, then you can explore the data set for possible trend, and that will design your query or design your question. Or you can, as I mentioned, generally search in the databases mentioned above, like Gene Expression Omnibus or SRA or bioproject databases for host response or human host response for SARS-CoV-2 infection. So this paper has a data set associated with that, so we can see the accession number and GUIDs, so we can get through the GUID. So this data set in the Gene Expression Omnibus data page will give you some basic information about what the data is all about, and what was the study design, and what was studied, and what are the samples involved, and what are the different replicates of the samples that we can find. So, and this also gives you access, or this also gives access to processed data. So we are not going to perform a detailed analysis of the processed data, but the processed data is an excellent place for you to start looking for the trends that will answer your queries. So when you take the processed data and then quickly perform a principal component analysis to study the sample distribution across the transcriptomics data in this case, then you can, if you can find such trends which can clearly separate samples that are of control type and samples that are given or that are subjected to treatment from RVA and RVC. So this trend gives you a lot of promise that you can specifically focus on, for example, responses due to RVA and responses due to RVC when compared to the control samples. So you have to perform such a basic exploratory analysis on the processed data that is available on almost all of the GEO data sets. So this could be a back and forth process. Some of the data set might not be conducive for a linear analysis like PCA, and some of the data set could be large, and you have to go through this process for a couple of more than one data set. That's what I meant. So this trend is promising. So when we see such a promising trend, so we collect the raw data that is also linked to the SRA, or short read assembly database, from this GEO database. All of these are from the one umbrella data repository, NCBI, database with NCBI. So we collect the raw data, raw FASTQ data, and then perform our own analysis or design our own analysis process for the raw data. So why do we do that? We can, for example, check if difference in a couple of analysis algorithms that we choose will change the data, or maybe we can take the raw data and perform our own normalization or processing or different processing methods. And most of the processing downstream analysis methods that you are going to employ will require the raw output from such a pipeline which is a sequence of analysis that you employ to the FASTQ or high throughput sequencing output. So we pre-process the data and then we map it onto the reference genome, for example, and we quantify the mRNA molecules that are mapped onto different genes. So this is a very short step-by-step process of transcriptomics analysis. This is just one example of variety of different analysis that you can perform on omics data. So this will actually give you, or this can actually result in a downstream analysis from which we can learn what are the genes that are differentially expressed when you compare two conditions. For example, in here treatment through one rotavirus strain or treatment through the other rotavirus strain compared to the control samples. So you can design your own analysis method that is different from the one that is published, or you can use an updated version of the human genome reference. So you have a variety of different reasons to perform your own analysis methods. So once you perform these analysis and get hold of structured data, then you can start exploring the data for the possible patterns and well-defined patterns. We already had a hint of the trend that could be explored from the data, and then these pattern exploration or these data exploration will tell you the necessary transformation or normalization that you need to perform so that you can use variety of different downstream analysis that will validate your hypothesis, or that will throw light on the question that you are working with. So yeah, so then you can perform in-depth analysis on the structured output. You can perform, for example, you can identify different genes that are differentially expressed, and then from the genes you can identify the pathways that are enriched, or you can identify which specific network, that is biological pathways and network of genes that is involved in characterizing this response, for example. And then we can also study various different biology of the sample, like if your data involves response to a drug resistance or treatment resistance, then we can also study the basic behind this development of this resistance from this analysis, for example. And at last, you will have to, after performing the data analysis and after performing the biological implications, you have to string all of these in one good story or one good project that will link what you have done and how you have done it, and what you infer the most from the variety of different analysis that you have performed for the project. So yeah, this, and then once you have completed or compiled the project report, then you can publish that in our Learn OmicsLogic portal, and yeah, and that will be public to many people, and you can share it with your peers. And then yeah, you can also, if you create a poster like this, you can also share those posters in the symposium or in several meetings that you are going to attend. So next up, here are some of the latest or some of the projects that are developed in our platform, and which are part of the research fellowship program from various different specializations. So we have, for example, in this case, this is a poster for a project done in collaboration between Noguchi Memorial Institute for Medical Research, and this poster is on malarial parasite Plasmodium falciparum, and its antigen, which is an antigen with a rupture neck protein binding binding. So the interactions, sorry. So this interaction is extremely critical for this for the falciparum to invade into host human response, host red blood cells. So different malarial vaccines are actually that are known to prevent such binding and to prevent this interaction, and these different candidates or these different vaccines have variety or variable success or variable successes at different stages. So this study actually performs a haplotype analysis or network analysis of genomic sequence data from the variable infection rates, and it compares with the data that is from the repository from another region. So by mapping on to the genomic variants that one identified from this haplotype study onto the protein structures, this paper or this poster actually concludes that there are some local variations and there are some epitope affecting variations in geographical locations that are different for different malarial parasites, and that needs to be taken into consideration while developing vaccines for malaria in a bulk. So and also you can also publish your results in bioRxiv preprint. Then you have paper or almost a near publication ready output that is available in the public, and you can link it to your ORCID ID, and then you can also share it with your share it with your peers, or use it for your future endeavors. And yeah, bioRxiv is one of the best way to store your almost ready projects. So there are other ways to do this also. So you can also develop your profile, and then this is from Sina Dolaner, and who finished her projects and she was able to work on leukemia and identify differentially expressed genes and protein coding genes that are involved in biogenesis and of these leukemia cells. And she also identified several non-coding RNAs and that are found to be significantly expressed, and this culminated into a research paper. So you can also publish your research if your research fellowship yields a very promising results, or if this could set the stage for further analysis that could result in a very promising paper.